Hi, in this lecture we're going to talk about enzymes you can use in a lab to modify nucleic acid, DNA or RNA. Those enzymes are super important when we try to clone our DNA or work with the DNA different experiment. So what kind of enzymes we're going to discuss? From polymerases to ligases, kinase, phosphatases, transferases. As you see, I'm not going to discuss restriction and nucleases because we're going to discuss a different lecture. Let's start with DNA polymerase 1. As you see, polymerase 1 is known to be involved in replication and in repair. And the major mechanism is that using a DNA or RNA primer, which supply the 3-OH end for the enzyme, it can incorporate different nucleotides if it has a DNA or RNA template. However, it's not just polymerase the DNA, the newly synthesized DNA strand from 5' to 3' direction, but also can degrade the DNA in both directions. So it has, we call the exonuclease activity, 5' to 3' or 3' to 5'. Now, what are those activities? Let's start with the 5' to 3' exonuclease activity. So there's the same direction as the replication. So what it can do, it can degrade a hat. So for example, it is some other already synthesized molecule, DNA molecule on this strand. It can actually cut it back. So for example, you have a primers downstream. You could actually cut it back and you can resynthesize that part of the DNA sequence. How about the 3' to 5' exonuclease activity? That's actually the different direction. So it's cutting back from the 3' to the 5' direction. And that's called the proofreading or error correcting activity. This is what happens during replication. A wrong nucleotide incorporated into DNA. Just a note, it can also slowly degrade the 3' end of the primer. Okay. So again, the polymerase 1, you can make DNA in the lab, okay, and usually using DNA template. If you have an array template, you have to use something called reverse transcriptase, so that's important. Again, needs primer and free nucleotides, 3-phosphate, DNTPs, and if you don't provide any nucleotide, you actually activate the exonuclease activity. Okay. If you provide one nucleotide out of 4, the exonuclease activity actually gonna kick in until this nucleotide needed in the sequence. All right, and that can be used for labeling. It's very interesting. All right, later I'm gonna explain a little bit more. So obviously in the lab, one of the main purpose we're using polymerase is in PCR reaction, and the famous one is Taq polymerase from a bacteria Thymus aquaticus, which lives in these hot springs. The ideal temperature for Taq polymerase is 72 degrees, and that's why we're able to use in our PCR reaction, since every PCR cycle starts with a high temperature to denature DNA, and that doesn't inactivate our Taq polymerase. So usually what we use in the lab is called the Taq polymerase for PCR reaction. However, I have to mention it that it lacking a very important activity called the 3' the 5' exonuclease or proofreading activity, which means it makes mistakes. It doesn't have this error correcting activity. Also needs high concentration of DNTPs, needs magnesium, very important. Of course, needs the free hydroxyl ends. So again, like other DNA polymerase, it needs a template and it can direct the DNA synthesis. So as you see, if you look at the half-life at 75 to 80 degrees, for example, for half-life at 95 degrees, 1 hours and 1.6 hours. At 37, however, it's about 10% this activity. I have another table showing the different different activity at different temperature. Why we using this? Even the fact that it can make mistakes, we're using it because it's cheap and not fussy. It's really one of the easiest enzyme to work with. So it was discovered, by Kerry Willis. He got Nobel Prize for it. The whole story. He went on A1 in California and motorcycle, and this whole idea how to fix those PCR reaction which he didn't have this thermostable enzyme came to him. However, I mentioned in my lectures that he just before he attended the lecture talked about this bacterial species in a hot spring. So this is the whole idea came together to him. So, but if you want to make a DNA strand, and the fidelity is very important. For example, if you make an expression vector and you want to express your DNA to protein, you need to use thermostable DNA polymerase, which has 3' to 5' exonuclease activity. And this is a few examples. My favorite is Vent. Half-life is about 7 hours. And also it is not sensitive to so-called PCR inhibitors. What are those PCR inhibitors? If you work with clinical samples like blood, biopsies, or any other fluids, we always have hard time to amplify the target genes, and mainly due to that very difficult to clean those samples for certain inhibitors. So lot of times we have to spend a lot of time to figure out how to remove certain inhibitors. For example, from blood, you remove some hem. From other tissue, you might have to remove some agents which blocking the enzyme reaction. So for that reason, even with the best DNA purification method, sometimes the PCR doesn't want to work. We found that Vent actually less sensitive to this PCR inhibitor, so I recommend to use that. Few is a fussy enzyme. Why it has very low error rate, is not easy to work with, and it is expensive. However, certain reaction like the side-directed mutagenesis has some advantage. It doesn't replace. So when we in side-directed mutagenesis, when we have this mutated primer for our reaction, it doesn't replace those primers. So it keeps the mutation in the reaction. So that's why we're using it. And the other one's a TGO. This is another thermostable DNA polymerase, a very low error rate and very sensitive and sensitive. So it is also people like it. Now here is the half-life at 95 degrees. Look at some Vent enzymes, especially Vent, a little bit expensive, but look at the half-life, very robust enzyme. One thing I want to mention, that those enzymes leaves the DNA after making the new DNA with different ends. Look at the Taq polymerase. Taq polymerase actually puts an A on the PCR product. It means if you want to use, for example, some kind of ligation reaction, you have to think about it, either blunt it or do something else with this A. And as you see, like deep Vent and Vent leaves it blunt, so you can use it as a blunt end fragment in the next reaction. So it's always important to keep in mind. As an error rate, as mentioned to you, that any kind of polymerase, like the Taq polymerase, doesn't have the 3' to 5' exonuclease activity, gonna leave lots of mistakes. So for example, it can leave like one mistake every 1,000 base pair, which is quite a bit if you think about it. If you have to make a protein from, let's say, a 2,000, 3,000 base pair fragment, you're gonna have 2 to 3 mutation just because the Taq polymerase. So think about that. Reverse transcriptase, as I mentioned, if you have an RNA as a template and you want to make DNA, you have to use a different enzyme. It's a polymerase, okay? again prefers template, RNA template. It also have an adenine H activity, which means after creating that DNA, it can digest the RNA template, which is very handy actually. Requires also primers, as I mentioned, RNA, very similar architecture than other polymerases. It shows a reaction, so you have your mRNA. Remember, mRNA a lot of times has this called the poly A tail. So one of the easiest way to prime this reaction to use oligo DT, as you said, T primer, which can prime this reaction and can make the DNA for you. Now, saying that, when you do reverse transcription, you can also do random primers. They're small primers, which is randomly prime different segments of your mRNA, or gene-specific primers, which bind very specific sequence on the gene. So you can prime it a lot of different ways. I usually like to prime it with random primers because it can you see the 3' end of your RNA molecule, and if I'm interested in the 5' end of my segment, sometimes if I prime it with the oligo DT, I don't get a good representation for the 5' end. So that's why I usually prefer random primers, or sometimes gene specific primers. That's also very, very good. All right, so let's go back to polymerases. How to create a blunt end? A lot of times we need to have our DNA blunted for the next reaction at the end. Remember, those excision endonucleases can yield fragments with either 3' or 5' nucleate overhangs. So you can use polymerases to either fill in. For example, if you have a 5' overhang, you could use a 5' to 3' polymerase activity to fill the blunt ended, or reverse, if you have a 3' overhangs, you can use the 3' to 5' X nuclease activity to chop it back. Let me show you some pictures, it's easier to understand. So here is, you have a 5' overhang, and as you see, using the polymerase activity, you can fill it in. Or you have a 3' overhang, okay, like here. You can use the exonuclease activity to chop it back, all right? So as you see, like for example, EcoR1 digestion gonna give you the 5' overhang, KPM1 other excision endonuclease giving you the 3' overhang, and depends on what you're gonna use. You can either fill it in or chop it back. Now I have to mention Clino fragment because we use this in the lab all the time. It has DNA polymerase and 3' to 5' X nuclease activity, but doesn't have the 5' to 3' X nuclease activity. So it has proofreading, but can chop ahead. All right, how they did it? Actually, what they did, because DNA polymerase is a holoenzyme, they removed those domain when it provides this 5' to 3' X nuclease activity, and they left called a large fragment or Clino fragment. That's a name. This is actually how you buy it in the catalog. You look for Clino fragment. So Clino fragment is very useful for a lot of different tasks. Again, it can synthesize DNA obviously from single-stranded templates. Typically, you need a free, again, the hydroxyl group. You need the primer. So you can make here is the little diagram. You can make this strand. Okay. So what else I can do with Clino? You can fill in the recessed 3' ends. Okay. For example, it shows you here. Okay. So this is a recessed. So you see recessed 3' end, and it can fill it in. Okay, so you can create a blunted. All right. So this is I said recessed 3' end. I could have said it's practically 5' overhangs. The same thing. Or you can digest away 3' overhangs using Clino. Okay. So what you can do is remember, if you have a 3' overhangs, you can kick in the 3' to 5' X nuclease activity. Okay, So if you add nucleotides, it practically gonna balance the X nuclease and polymerase activity. So the minute is reaching the part when you have like right here you can see double strand, it gonna stop right there. Okay. So how can you use for labeling? So if you think about it, can we label using this enzyme either with radioactivity or biotin or some other ways, other labeling molecules? Yes, you can. Okay. In that case, however, so if you want to incorporate those radioactive nucleotides or biotin labeled nucleotides to label your DNA, practically what you have to do, you have to provide at least one of the 4 nucleotides that's labeled one, and you can incorporate it to those very frequently. Use this reaction. Now, I had to mention that we have something called the exo minus clino fragment. That one actually doesn't even have the 3' to 5' X nuclease activity, and this because in some reaction this activity actually not required. So just let you know, depends on what exactly you want to know. You have to pick the right fragment. Now let's go back to some labeling methods, and one of the famous one called the Nick translation, and you can even buy a kit, and it's using polymerase 1. What's happening here, you start your DNA, and usually they use a low amount of DNA as one just to make this nick in one of the strand, and after you add DNA polymerase 1, for example, and you label this upper strand. Don't forget, polymerase 1 has the 5' to 3' X nuclease activity, so it's gonna chop ahead, yes, and replace it with some labeled nucleotide if your nucleotide are labeled, and that can produce a very high labeled either radio labeled oligonucleotide, biotin labeled oligonucleotide, which can be used for the next experiment. All right, I'm not gonna go. Obviously, the DNA sequencing, the Sanger sequencing is also using the whole idea that we're making a second strand of a single stranded template, and remember, it is also incorporate certain dideoxy bases, which has termination the polymerase reaction. Please review the Sanger method. I covered in other part of this course. Is remember, it's still used very heavily, so I want you to know it. All right. So the next question is, so what other polymerase we can use in a lab? Now, sometimes you want to make RNA certain experiment, and we have RNA polymerases. So those synthesize RNA using DNA template. What they need, as we mentioned in a transcription class, they require promoters with very specific sequences. Okay, and remember, for example, E. coli RNA polymerase has all those 5 subunits plus the sigma factor, and remember, it needs to recognize those thousands of E. coli promoters. For that reason, in the lab, we're using bacteriophage polymerases because they have very specific sequence they like to bind, and those sequences can be incorporated in our vectors. So T3, T7, T3, T7, SP6 polymerase. This is the one we're using in the lab. Again, they need a promoter in your construct, but if it has it, and after the promoter sequence you have a DNA sequence, it's gonna make RNA for you. I love those enzymes. They are very efficient enzymes. I never had problem with them. All right. So remember that you can make RNA from your vector if you have those promoter sequence. The ligase, obviously we learned about ligase, very important in a lot of different reaction, replication, repair, even recombination. We know joining linear DNA fragments together with covalent bonds. Okay, they need obviously a hydroxyl end, 3' hydroxyl, and a 5' phosphate end. Okay, so here's the reaction. This is the T4 DNA ligase, one of the ligases we use most frequently in the lab, and looks like is in this reaction it is annealing a EcoR1 sticky end. Both DNA fragment has the same end, so it will be an easy reaction, as you see with the hydroxyl group, with the phosphate group, and it's able to ligate together those two ends. Okay, very efficient if you're working on sticky ends. However, it can also work on blunt ends. Needs ATP. That's very important. So please be sure that your buffer contains ATP. Again, depends on you have sticky ends or blunt ends. You need less or more ligase in your reaction. Timing is also important. It shows for example here some fragments. We want to ligate them together. As you see, 2 units of enzyme. You can already see the shift, which shows the ligation reaction in 5 minutes. But as you see, if you go down, like let's say there's a 0.02, you need even 45 minutes doesn't produce the same reaction. So it's very important that you use this right amount, and you use right amount of enzyme with the right amount of time. The other thing is very important is the temperature. Usually it is between 12 or 16 degrees. Sometimes I like even 4 degree overnight. Depends on what exactly you will, how efficient is your ligation reaction. If it's a blunt end ligation, I recommend a longer time, more enzymes. If it's a sticky end, it's gonna go within very fast, even as you see in 5 minutes. One thing that the companies mention that ligases can have some X nuclease activity, so that's why they always recommend to lower the temperature, because that lowers the X nuclease activity. Right, and here's some example for ligases. So you have the E. coli DNA ligase. Unfortunately, it only works on sticky ends. But if you're using the T4 ligase, it works both sticky and blunt ends. Actually, they have a T4 adenine ligase if you want to put together adenine molecules. I never used that, but I heard about that. Now, the next thing we need to talk about kinases and phosphatases. Just remember, kinase can add phosphate group, the phosphatase can remove it. So this is practically, as you see, it can complement each other. Kinase needs ATP. Okay, that's very important. And when you add ATP, if you want to label it, because it actually takes this part of the phosphate, the gamma phosphate. So if you want to use kinase for labeling, if you have a label on the gamma phosphate, you can actually use it. This is not sequence specific, so you can practically do any kind of DNA sequence. All right, so let's start with the phosphatase. Famous one is alkaline phosphatase, or AP. This remove the phosphate group from the 5' end. Okay, now what we use it for? Most of the time we use it to prevent ligation. Okay, so if you treat the DNA fragments with AP, you cannot ligate it after, because it's missing the phosphate group. Okay, can be used also for labeling. You will see later. All right, so a famous one is the bacterial alkaline phosphatase, active, but after it's very difficult to heat inactivate, and that's a problem, because you want to remove after phosphatase in the reaction. Calf intestinal alkaline phosphatase, or CYP, people like it, but it's less active, but you can easily deactivate it 75 for 10 minutes, or use some protease K enzyme to take it out. So that's actually a better way to go, because most of the time you have to take out this phosphatase. Shrimp alkaline phosphatase also is heat sensitive, and use it a lot of times for dephosphorylation of some DNA molecules. So this is for example, showing you how to the shrimp alkaline phosphatase, how to remove those 5' phosphate group. Actually, on this one looks like the calf intestinal one. But again, if you remove this phosphate group on the 5' end, you see you cannot ligate it anymore. Okay, so why it is important? Think about it when you try to put something in a vector, a piece of foreign insert. You don't want the vector close on you. So if you use phosphatase and remove the 5' phosphate from the vector cannot ligate itself back, and you have much less background. Let's go back to kinases for a second. So this add phosphate group, now what it needs ATP, of course magnesium, everything needs magnesium. Okay, so it's interestingly that if you add ADP, a kinase can exhibit phosphatase activity. I remember that when I was looking first time I got this, the instruction how to use kinases, and it was like small prints talk about that also has some phosphatase activity in certain conditions. So be sure that you understand how to work with those enzymes, because it can surprise you. So what we use it, if you have an oligonucleotide, okay, you could add phosphate group at the 5' end, and again if you're using labeled ATP, it's gonna be labeled. You can also add the 5' phosphate to allow them to ligation, and you can also do some remove some 3' phosphate group, so it has some phosphatase activity. So just be sure that you use the right condition to be sure that you using the right activity for kinases. The famous one we use in the lab is called the T4 kinase. All right, let's talk about a few nucleases. We always, talk about DNA as one, but let's see what other nuclease available. So nuclease BAL31 is an exonuclease. Interestingly, it can degrade both the 3' end and the 5' end. It works on double-stranded DNA. Okay, it will not make any kind of nicks. Okay. But if you have an internal nick, it actually can cut it back. The only problem working with BAL31, it's not, so when it's chopping back, it's not 100% blunt ended. So it makes sort of a ragged end. After you have to, maybe if you need really a blunt end, you have to fill it in, for example, with T4 polymerase. Okay, so this is how it works. So here's the duplex, and as you see, it chops it back. Not 100% unfortunately blunt end. If you have a nick, can also chop it back. Again, problem is not 100% blunt end, so you have to think about that. The other one is called the Mung-Bean nuclease. That's a single strand specific DNA or RNA nuclease. Okay, so it cuts back a single, if for example here, see you have some overhangs, it actually can able to cut it back. All right, so practically you're making blunt end. So that's one of the ways you can make some blunt end molecules. Okay, nuclease S1, that's what we use a lot in the lab, because it actually degrades single-stranded nucleic acid very, very effectively, very efficient on DNA. Okay, and very interestingly that we use it for some opening some heart pin. Let me show you some heart pin loop. Again, it can remove 5' and 3' overhangs on the single-stranded DNA. So let's see. So here is we call the S1 nuclease protection assay. S1 nuclease protection assay was used if you have a probe, okay, a probe, for example, for a genomic DNA segment, and you want to map where are the exons and how big are those exons. So if you have the undersized probe, which only covers, let's say, exon 1 and exon 2, okay, you could make it double strand, as you see with the probe. So you can let the probe bind to your segment. Now what happens is the intron gonna loop out like that, okay? And you can come back with S1 nuclease and chop it back. So what happens after? Now you're gonna have two fragments, okay? You're gonna have exon 1 and exon 2 fragments, and you can see the size of it. So this is how you can practically map it, okay? The other one of the famous example to use S1 nuclease, if you like, for example, you make an mRNA and synthesize a DNA RNA hapid, a lot of times you have this hair loop in your reaction, which is not bad because after you can add clino and some DNTPs and you can make the second strand, and to remove those hair loop you can use S1 nuclease. So this is one of the things we do in the lab also. Okay, now back to a little bit you can also you can use S1 an RNase protection assay, because it is also gonna get rid of some RNA. So here's what you have. So you have your mRNA, okay, and you have an antisense probe again, against your mRNA. You will see this gonna bind here, but after, if you digest with S1, this part and this part is gonna be not represented, and you can only gonna see the part which is bind to your mRNA. So this is also can find whether your sample has those sequence you're looking for. Okay, now next one is exonuclease 3. I like exonuclease 3 because what it does, it removes the nucleotide from the 3' hydroxyl end. But what's good about it, you can time it, so it's very stepwise removal. So it has actually a table. You could say for 10 minutes remove that much, 20 minutes you remove that much. Could be very handy if you want to chop back your 3' hydroxyl end, your DNA, and you want to know how much to chop it back. Okay, so after you can either leave a blunt end or 5' overhangs, or of course also can leave some internal nicks. Okay, now so it needs a duplex DNA. The only things that if you have a 3' overhangs, it's not gonna work. Okay, it is also inactive on single strand DNA. So you need a duplex DNA. But again, why would you use this exonuclease 3? Because you can almost time it how much you want to cut it back. So it could be very, very handy. All right. Now, before we mention DNase 1, this is from bovine pancreas, usually the isolated. It practically depends on what kind of iron you provide. If you provide magnesium ion, it usually gives you some nicks. Remember, there's a nicks we can use for nick translation. It's interesting, if you provide manganese ion, it starts to cleave both strands at the same position. So it's very interesting how depends on what you provide. It can either just cause a nick or cleave both strands at the very same position. However, it leaves a regidence. Okay, so what do you use it for? I mean, most of the time it's just really use it for just eliminate DNA from preparation. For example, if you make RNA and you don't need your DNA template, you can use DNase 1. You can buy RNase-free DNase 1. Also footprinting, we discussed footprinting before. I'm gonna go back for a second. The main enzyme is, and as I mentioned before, it can nick the DNA prior to nick translation. So just let's go back for footprinting. Footprinting is practically you have to label your DNA, okay, and after you have to add DNase. DNA is gonna start to introduce those nicks. So you're gonna have different fragments. However, if you have a specific binding protein, right here, for example, cannot bind, cannot nick the part with the protein cover the DNA, and then in that case some fragment will be missing. So if you have some control, you can actually know exactly what fragment is missing, and you know where is your protein bind to your DNA. It's very important for any kind of promoter studies, transcription factor analysis, where they're gonna bind to your DNA fragment. All right. So, but now let's say you have a DNA prep and you don't want RNA in it because it's gonna contaminate your reaction. So fortunately we have ribonucleases, unfortunately quite a bit. Remember, even on your skin you have a lot of those ribonucleases because it's protection for you from RNA viruses. So RNase A, practically it removes single base mismatches, okay, from RNA DNA, RNA hybrids, or can chop single-stranded RNA. All right, usually cleaves to 3' and to C and U. All right, so again, if it's some kind of mismatch, it's not gonna cleave at exactly the mismatch site, or just if you have a single strand RNA, it's gonna just chop it back. So what we use it for? So lot of times any kind of RNA contamination and preparation, like a plasmid DNA or any kind of DNA prep, you can also map some mutations. As I mentioned, if you have a mismatch, it cuts a mismatch, so you can identify if you have a mutation in your DNA or RNA molecules. Okay. The famous one is RNase. Is most of the time you use it in the lab. Very easy enzyme. Actually, it's too easy. You really have to be sure it's not contaminating your lab. RNase T1, there's another enzyme. Sometimes they have this like a mixture, because that one is cleaves at 3' to G residues. So you can sometimes buy RNase A and T mixture. In that case, you H. Sorry, I got tongue tied. It's very important to remove the RNA from RNA-DNA hybrid and reverse transcriptase. Most reverse transcriptase actually has RNase H activity. If your reverse transcriptase doesn't have, please add some RNase H into your reverse transcription. All right. The last two one is one of the terminal transferase. I worked with terminal transferase. I love terminal transferase because it can add nucleotides to the 3' end, and it can add a lot of nucleotides actually. Okay, as many as several thousand, and you can even see, it depends on what you want to add. If the nucleotide is purine, you have to use magnesium in your buffer. If it's pyrimidine, you have to use cobalt. Okay, so it works on single strand DNA, or a double strand DNA. It has a 3' overhangs. Okay, so you have to know what exactly your DNA looks like before you use it. Okay, the best, if you have a 3' overhangs, and actually that's the best. The blunt, if you have blunt or recess, it will work but less efficient. Okay, so let me tell you what I used it for. First of all, you can label the 3' DNA. You can add a labeled nucleotide, but you can also add, for example, here, let's see, you have your DNA, and you can add, for example, lots of T's. And you will say, why would I add a lot of T's? Let's just see if I, okay, it was a little bit different because it added G's. But if you, let's say you have a plasmid and you have a cDNA, and you want to make a cDNA library, and you want a very efficient way to ligate your plasmid and the cDNA fragments, what you could do, you could add C's on your cDNA, and you can add G's on your plasmid, okay? And after you can ligate the two extremely efficiently. So that's what I was used when I did some cDNA libraries, because in cDNA library you need a very efficient ligation reaction, because you have lots of fragment, lots of plasmid. So you want to be sure that all the cDNA fragment you created with reverse transcriptase gonna be represented. So this is what you could do. Okay, so this is extremely efficient and extremely easy. And the last enzyme is the methylating enzyme. So the one we usually use is the methyltransferase. Practically it methylates the adenine residues. You know, we talked about that from bacterial system. And if you methylate your DNA, it's going to prevent the cleavage of some restriction enzymes. The other one, if you want to methylate on cytosine, we have the DCM methyltransferase. This is again a bacterial enzyme, and it can methylate the cytosine at certain sequences. All right, so that's it. I hope now you have some appreciation for all those different enzymes. It can be really useful in the lab when you manipulate your DNA for a different experiment. The bottom line is probably you have some now appreciation that you need to know what kind of what you have, what kind of end you have on your DNA or RNA to be sure that you know that you using the right enzyme. Thank you.